Question 10 says a student investigates the factors affecting the rate of photosynthesis. Complete the balanced chemical equation for photosynthesis. That will be carbon dioxide plus water makes glucose, which is C6H12O6 plus oxygen. And then you balance it by adding a 6 in front of everything except for the glucose. So there's 6, 6, and 6. And that's done. And just a quick point, if this was equation for respiration, then it'll just be the other way around, right? It'll be 6 oxygens, add glucose, make 6 waters and 6 carbon dioxide. Now, before I proceed, I want us to just revise the factors that affect the rate of photosynthesis, because that will help us for the rest of this question. There are 3 factors that affect this. Number 1 is, in fact, this equation gives you a clue, is the CO2. Now, don't say amount of CO2, volume of CO2, is the concentration of CO2 that affects rate of photosynthesis. The second one is, think, what does photosynthesis require? It requires light and chloroplast, right? So light intensity is essential, and that will definitely affect photosynthesis rate. And the third one is temperature, and this will become important as we go down. So these are 3 factors. Okay, so here's a setup. Let me zoom out a little bit so you can see the whole thing. Right, so what does he actually do? He has this setup here where, first of all, step 1 is he fills a test tube with 5% sodium hydrogen carbonate solution. So there it is. And if you're clever, you will realize that this is actually the source of CO2. So this is the carbon dioxide that is in the photosynthesis equation. So that comes from here. Then what does he do? He places the pond inside. There we go. And then he places the lamp 5 centimeters away from the test tube. 5 centimeters away from the test tube. And then he counts the number of bubbles. What are these bubbles? What are these bubbles? Look at the equation to tell me what are the bubbles coming out of the plant. Those bubbles, those green bubbles that I'm highlighting right now, that is the oxygen bubble, right? And as I've said just now, the carbon dioxide comes from here. The carbon dioxide comes from here. So just color code it in your mind as you go along. Okay, and then he counts the number of bubbles, which is the oxygen produced in 2 minutes. And then he repeats the whole thing for the following distances at 10 centimeters, and then 15, 20, and then 25. So he keeps moving the lamp away, like farther away from that. And why is he doing that? Because he's basically changing the intensity of the light, right? Because the farther away you move the lamp, the less intense will be shining on the plant. So from this, you should already conclude that the independent variable, all of these here, that is the independent variable. That's what he's changing, the independent variable. So as you're reading this, you should sort of be building up these key points in your head already. And then he repeats the whole thing from 5% to 10%. So that's what he's doing. And this table here shows you his results. So he moves the lamp farther away, and then he counts the number of bubbles of oxygen in 2 minutes for each of those distances, and then he repeats it at 5 and then for 10% sodium hydrogen carbonate. Okay, now it's asking, the student used the same color of light in the investigation. Describe how the student could control one other relevant named abiotic factor. So abiotic, you guys remember, this means that it is non-living, and these are things like temperature, pH, and that's exactly the case here. The other way to think about what he needs to keep the same, the control variable, right, is to come back up and look at my checklist just now. Have a look. These are the 3 things that really affect the rate of photosynthesis. Your rate of photosynthesis here, that is clearly the dependent variable, isn't it? That's what you're measuring, the dependent variable. So what's he changing? He is changing the light intensity because he's moving the lamp away. So this is the IV, this is the DV, and he's also changing carbon dioxide concentration. He had it at 5% and 10%. Remember, he repeats it at 5 and 10%. So that's like the other IV. There's 2 IVs in this experiment. So Therefore, the last one must be kept constant. It's got to be a CV, a control variable. Otherwise, your experiment would not be fair. So it's good to know these three in your head in advance because you're thinking, are they controlling for temperature? Are they controlling for temperature? So temperature is definitely a point here. So temperature, and it says how, and make sure you don't miss that point because it's 2 marks here, guys, is that you have to say how. How do you control for temperature? You use a water bath. Don't say thermometer. That doesn't actually control. That doesn't keep the temperature the same. The thermometer just measures the temperature. So it's a water bath that keeps it the same. Now, another point is you could also say pH. You can say keep the pH the same. And how do you keep that the same? By using a buffer. Use a buffer. So you could choose either of these bullet points, but for each one, you must mention both points to get the 2 marks. Okay, next question. It says, explain the effect of changing the distance of the lamp on the rate of photosynthesis of the pondweed in the 2 different concentrations, right? So there was a 5% and there was a 10%. So basically explain this entire, make sense of this whole table, his results. Doesn't make sense. Why does it look like that? What I've done here is I've plotted a graph to make sense of the data. Now, you're not asked to do this on the actual paper, at least not for this paper, but a graph really shows you how to understand what this experiment is telling you. Okay, now I've plotted the independent variable, which is the distance of the lamp away from the test tube on the x-axis, and then the dependent variable is the number of bubbles that they're counting, and of course they're counting it at 5% and 10% concentration. So that's the 2 colors, the blue and the purple, right? Now when you look at this graph, I want you to think about 2 things you're going to explain. Number 1 is the overall trend of both graphs. And then number 2 is the changing point, like what is different and how to explain that. The changing is, let's call it turning point, the turning point. So overall trend, just looking at this graph is, let's zoom in a little bit, okay? So overall trend, looking at this graph is, as the distance of the lamp increases, means farther away from the test tube, overall, it's a negative trend. It's a negative trend. So I would put that down. I said increase distance of lamp, and explain that, right? The question was explain. So you can't just describe like, oh, this increase and that decrease. You have to tell me why. So as the distance of the lamp increases, then the light intensity drops, right? That's common sense. The light intensity drops. And if the light intensity drops, then of course you would expect less photosynthesis and therefore less number of bubbles. So less rate or reduced rate of photosynthesis. So that is entirely in line with logic. So that's the overall trend. Now, what about the turning point? The turning point, guys, is where? It's like this bit here, isn't it? Because if you look at it, if I draw a dotted line here, before this dotted line, the blue and the purple is different. But after this dotted line, look, they're both going down. So why is it? Because this is definitely worth explaining, right? This question is 4 marks. So we have to make sense. So before the turning point, just try to understand. Look, the light is really close at 5 centimeters. Just 5 cm away from the test tube. So I would expect to get a lot of bubbles produced because that should be a lot of rate of photosynthesis, isn't it? However, what's happening for the purple line, which is the 5%, remember, I'll put that down here. The 5% is the purple line, and then the 10% is the blue line. Okay, that's the 10%. So that's 10%. So why is it at 5% is nothing happening here? I should expect the purple line should be like this, isn't it? Because if it's closer to the test tube, then I should be getting more bubbles. But I'm only still getting 30 bubbles, which is the same as 10 cm, which is 10 and 15 cm. Why? Whenever you see this flat line on rate of photosynthesis questions, you have to think of this point called the limiting factor. Something is being a limiting factor. It means that it's preventing there to be more more bubbles being counted. And if you go back to what we just mentioned, the 3 factors that affect rate of photosynthesis, right? There is carbon dioxide concentration, light intensity, and temperature. Temperature we're keeping the same. Remember, that was one of the control variables. Light intensity we are changing here because we're moving the light away. And what's the last one here then? What is stopping the purple one from being higher up here? It's the third thing. It was the concentration of carbon dioxide. Yeah, because temperature is the same, and I am already changing the distance, which is the light intensity. So the only thing that's not changing in this purple one is the carbon dioxide concentration. So the reason this is a flat line is because the carbon dioxide concentration is limiting it, So that's what I'm trying to say here. Here is CO2 concentration is a limiting factor, It's not allowing the bubbles to be more at the beginning. I would expect this point to be up here, isn't it? So so is a limiting factor. And if you're still not convinced, just look at the blue. Look at the blue one now. The blue one is subject to the same distance from the light, right? Also 5, 10, 15, but it's much higher up here. And what's the difference for the blue and the purple? The blue is 10% concentration CO2. So that should convince you, right? When they're both blue and purples, both at 5 cm away from the lamp, but at 5%, you only get 30 bubbles. But if I boost up the carbon dioxide concentration to 10%, then I'm counting extra bubbles. I'm getting up to 50 bubbles in the 2 minutes. So that proves to you that this bit, CO2, is being a limiting factor. Okay, so we've explained this whole part here before. So that's the first point here, is before the turning point, the CO2 concentration is limiting the blue line. Now, what about afterwards? What about here? Here, they're both dropping. So how would you explain this bit? Why would rate of photosynthesis go down? Because look, this is really far away now, isn't it? This is like 15, 20, 25 centimeters. That's quite far away from the test tube. So the lamp is way too far away. And if it's that far away, then what's limiting the rate of photosynthesis now, since the lamp is so far away? So this bit is when the light intensity is limiting it, because it's just too far away. So light intensity limits, or is the limiting factor, limits further rate of further increase in rate in photosynthesis, basically photosynthesis. So that's what they're expecting from you. The last reminder is that to use the actual numbers in your answer. So you would say at 5, 10, 15 cm for the purple graph, for the 5% graph, carbon dioxide concentration is limiting the rate of photosynthesis. And you would say beyond this point, so at 15, 20, 25 centimeters away from the lamp, then the light intensity is limiting the rate of photosynthesis. So you're always talking about limiting factors, get it? This bit is the CO2 that's limiting it, and this bit is the light intensity that's limiting it. All right, part 3 here says describe one way the student could modify this method to obtain a more accurate result. So when they're talking about accuracy, you want to think about what equipment you could use, like a better improved equipment. So instead of using what did you use? You just use a little test tube, right? Test tube doesn't have any quantitative measurement. If you ever look at a test tube in the lab, there's no markings there. So you don't get to measure anything. So that's actually not a very quantitative method. So what measuring apparatus has a measuring numbers on the side? The cylinder, right? Measuring, use a measuring cylinder, or even better, or burette, right? A burette, because those have markings. You know, burette has got markings on it. Do not say pipette. Pipette don't have markings. Pipette just measures one fixed volume, but it doesn't measure volume as the bubbles are being produced. So that's one way you can do it. Another way is, do you remember how the person was doing the experiment? They were counting the number of bubbles. That's not very accurate, because sometimes you miss a bubble, right? Maybe you count too many, or you forget to see one because you blinked. So what's a better way to measure the oxygen produced? How do you measure volume of gas produced? You should do exactly that, measure the volume. So measure the volume, actually measure, quantify the volume of oxygen produced. And you can do that with the with the burette or the measuring cylinder. Next point is describe how the student can ensure the results of the investigation are reliable. Okay? So to be reliable, there's one key word you have to say. You have to repeat. Repeat, and don't just end there. Give me more. Repeat and calculate what? Calculate an average or mean. Yeah? And also remove anomalies from calculation, right? So when you calculate your average, you don't include the anomalies because those are too weird. Remove anomalies from calculation. So that will give you a model answer for reliability question. Okay, so question 11, it says biological washing powders contain protease enzymes to digest proteins in clothing stains. Now, scientists have produced recombinant plasmids contain a gene for a protease enzyme that works at high temperatures. Now, the plasmids have been inserted into bacteria so that the bacteria produce the protease enzyme. And it asks you to describe how named enzymes are used to produce a recombinant plasmid containing a gene for a protease. Okay, so let's go through this thing more wholesomely. So here is a bacteria cell that I'm drawing. And as you know, bacteria cells usually have a cell wall as well. Sometimes they also have a flagella. And then they have this, they're prokaryotes, so they do not have a nucleus, right? So they just have their DNA floating in the cytoplasm. And of course, they would have some ribosomes. And then they will have, most importantly for this question, are these circle things. And that would be plasmids. Okay, that would be the plasmids. Now, why is that important? Because I'm going to get the bacteria to work for us. How? I'm going to extract one of these plasmids. So let's take that out. Let's do that right now. So let's take that out. And what I do with this, first of all, I'm going to make it a little bit larger. Okay, so there's the very big plasmid. And what we do with it is that we're going to, first of all, cut a bit of it out. So once we've cut that big section out, then we're left with just this bit of the plasmid, right? Just this bit of the plasmid. Then we're going to put a section in, and this pink section is the thing I've highlighted in pink. That is this gene for the protease the protease enzyme that works at high temperatures, because this is the enzyme that we want, right? So I'm going to put the gene to make this enzyme into the plasmid. Now, after I put it in, I have to stick it, right? It's just like cut and stick. So I've done the cutting. Now I've got to stick these two bits back. And the way to stick it is you're going to use another enzyme to do this sticking. Now, the name for these enzymes here, the cutting enzyme, that is called restriction enzyme. Restriction enzyme. And the sticking enzyme, the green one here, that is called ligase. And those are the so-called named enzymes, the named enzymes that the question is asking for, the named enzymes, right? So what ultimately you're doing is you're going to use restriction enzyme to cut out a part of the plasmid, and then you're going to insert the gene that you want, and that is the pink gene that works, the enzyme that works at high temperatures, and you're going to stick it back with ligase. So you must have these two words to get those marks because it's asking for the name. And don't end there, but you have to put this recombinant plasmid. So this thing now, this whole thing is known as a recombinant plasmid because you've recombinant plasmid because you have put a pink gene into it. So you've recombined the black and the red gene, right? The black and the pink gene. So that is the recombinant plasmid. And just to finish off the story, you would put this back into the bacteria, and the bacteria doesn't know that you put a pink gene inside it, right? Because it's just reading a bunch of DNA like A, T, C, G, 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 that. So you just put it back in, and the bacteria will just do its usual protein synthesis business. And as it does that, it's going to naturally just make a bunch of pink proteins for you because it's just doing its own protein synthesis. And then you can then collect that protein and then harvest it and use that for the washing powders. And these are biological washing powders. So the key word for this is really the named enzymes, which is the restriction and the ligase. And obviously what they do. One is the cutting, one is the sticking. All right, part B says, design an investigation to find the best temperature for removing protein stains by using washing powder that contains protease enzyme. Include experimental details in your answer and write in full sentences. Oh, okay, right. So when I'm reading this question, the first thing is I've got to know my independent variable and my dependent variable. So my independent variable, that is, let's use a different color actually. My independent variable is the temperature. Right? Independent variable. That is what I'm changing. Independent variable. And then what is my dependent variable? That is what I am measuring, right? Measuring. So what am I measuring? I'm measuring how much or how well, how effectively it has removed protein stains. Okay? How effectively. So that is the dependent variable. So just reading the question, you have to be able to know these two already. And then you should also be thinking control variable, which is the things that are keeping the same. Now, you probably heard of the famous CORMS approach. So CORMS stands for C-O-R-M, and there's usually two M's, M1, M2, and then two S's, S1, and S2. Now, what these stand for is change, organism, repeat, measure, and then same, and same. Now I'm going to do a bit more color coding to help you. Now the one that you're actually changing, that is the independent variable. That is your IV. And what are you measuring? The thing you're measuring is your dependent variable. So this should sort of link a lot of things up in your mind already. Okay, so I'm going to just go all the way up like that. So link all these concepts up in your head. You're changing the independent variable. And now you should be able to start putting your answer together. So what you're changing is the temperature. Now, don't just stop there. Don't just write temperature. You must always give 5, a range of temperatures. So say you're going to use a range of 5 different temperatures. And you can even write out 5 temperatures. Of course, they've got to be feasible, you know? Okay, so realistic temperatures. 5 different temperatures. And you can even talk about using a thermometer to measure that. Thermometer and water bath and so on, right? Thermometer, water bath. So always think of equipment, apparatus, right? When you're doing experiment questions. So that's the change. What organism are we using? We are using enzymes. We're using enzyme. And when you're thinking about the enzyme, you have to think, especially for biology, the organism, unless that is your independent variable, otherwise you should be keeping that the same. So what does that mean? What are you keeping the same? Should be the same species, right? But it's not, enzyme's not really a living thing. So the same type of enzyme. What else is important about the enzyme? Same type, same mass. Do not say same amount, guys, because amount is too vague for a science paper. So the mass of the enzyme, all of these are important. So that's the organism. Repeat. So just putting that one word is a mark. But what are you repeating, right? So you're actually repeating whatever you're measuring. So really, the M should come before the R logically, but they want to make a word that sounds like something at CORMS. So what you're measuring is the dependent variable. So how are you going to measure this? You're going to measure how well it's removing the stains, right? So if you have a big ugly stain, like this, how are you going to measure it? How are you going to measure whether or not it's been effectively removed? You can, for example, measure the size of it. Size is too vague, so perhaps the diameter of it or the area, right? Area of stain after it's removed, or basically how much is shrunk, right? What else can you measure? You can also measure the color of the stain. So something, you have to think about what you're measuring about the stain. Color, darkness or darkness or paleness of the stain. Paleness of the stain. Now, the other thing you've got to measure is time. Time is very important because you've got to be using the same amount of time to to do this experiment at the 5 different temperatures, right? So you're going to wash them all for example, 30 minutes. You can't do that differently for the different temperatures. So time is the second thing that you're going to measure. That's the second thing you're going to measure. And now that you've got what you're measuring, you can start to say what you're going to repeat, right? You're going to repeat this whole method at 5 different temperatures, and you're going to repeat it 3 times and find average. Find average. So it's all coming together. Find average. Okay. And then next one down here is same. What do you have to keep the same? Now, there's lots to write down. Write down at least 3 to 4, because you might write one down that's not worthy of a mark. So put down as many as you can. Some worthwhile ones is think enzymes. What's going to affect the enzyme activity that you don't want it to affect it? One is temperature, but we're already changing the temperature, so that's okay. What else affects enzyme activity? What else? pH, right? You've got to keep that the same. pH keep same, and you can say use buffer. Use a buffer, because buffers, that's what they do. They help to keep the pH the same. What else do you have to keep the same? Look at your experiment, right? The stain, that should be the same type of stain, right? Like, are you removing ketchup or sauce or what? So same stain. Don't use ketchup for one and soy sauce for another. You got to use the same stain. Same stain. What else? Same type of clothing, isn't it? Same type of clothing. So just picture it. If you're running out of ideas, just picture doing this experiment and think of what you're using. You've got stain, you've got the cloth. All of these should be kept the same, because if they're not kept the same, then it makes the test unfair. So guys, this is how you do a quorums question. One last point that's very important is that it's an enzyme that you're using that is removing the stain, and the enzyme is a protease. It's a protease. And if you know anything about protease, is that they actually break down proteins, right? And therefore, your stain has got to be a protein stain. Does that make sense? So every detail is important. It's not just any stain, like there's not anything. So you've got to refer back to the question to see what type of stain you're using and write that down. You're going to use the same protein for the stain, in the stain, to be broken down by the protease. So yellow box, this is the balanced equation for photosynthesis. Please memorize that. It's got a big 6, and big 6 in front of everyone except for the glucose, and glucose is C6H12O6. And once you've memorized this, the good news is the balanced equation for respiration is just the other way around. So you only need to memorize one of them. The 3 purple things here, they are the factors that affect the rate of photosynthesis: CO2 concentration, light intensity, and temperature. Now, red box, whenever you look in a graph and something is expected to be higher, but it's not, for example, this purple zone, why is it flat when you would expect it to be higher, such as the blue one? Then you think of the possibility of limiting factor. And in this case, the limiting factor for the purple bit, the flat line bit, is the concentration of CO2. Okay, purple box here, this is a recombinant plasmid. Remember, to make that, you need 2 enzymes, the restriction enzyme and the ligase. The restriction enzyme and the ligase. And once you've made that, you end up with a recombinant plasmid. And finally, the black box, this was an excellent practice on quorums question. Just a reminder that your change, your independent variable, you need to list 5 different things for that range. And then make sure you measure and talk about exactly how you're going to measure. Is it the area? Is it the rate? Is it the time? Is it the mass? Give details of what you're measuring. And for same, write down at least 3 and choose the best ones. The same ones are the control variables.